Hi, this is my gut microbiome lab presentation, and we were studying the microbiome of the gut. But first off, a microbiome is a type and proportion of microorganisms living in an environment inside of your body. And gut microbiome are the microbes and organisms living in the environment in your digestive system, and they help promote healthy digestion within the body, or if there are certain bacteria that are in there that could hurt the digestive system. One disease that is related to the gut microbiome is obesity, and it can be correlated between that because the amount of bacteroids and firmicutes is related to obesity or not having obesity. If you have more of the bacteria, you could become obese, and if you have the normal amount, you're healthy. And they also affect mainly women and children with autistic spectrum disorders, and studies have shown that the gut microbiome and the mother may play a role in the occurrence of autism. And the relationship is because microbes found in children and women are related to the occurrence of autism spectrum disorders, and they found bacteria that related to the disease that was in the gut microbiome. They aren't 100% sure on how it got there, but there was a correlation, not a causation. And the hypothesis that we tested in this experiment was whether the sex of two similar dogs would have an effect on the bacteria found in the gut microbiome. So if males have healthier in dogs, or if females have healthier gut microbiomes. So first, when we started the experiment, we had to pick two subjects. So we picked two German Shepherd dogs, one male, one female, both around the same age, same weight, same health. They were both unaltered. And then once we picked our samples, we used the Microbiome kit to isolate the DNA from the fecal samples, and we followed the lab manual instructions for this verbatim. And next week, after we isolated the DNA, we then concentrated it to make sure that it was pure, and we isolated it tenfold using the commercially available kit, the Zymo kit. And the DNA is bound to a filter column in this process, and it cleans it from impurities, from bacteria, RNA, proteins. We follow the instructions verbatim for that as well, and we made 10 times more concentrated DNA than we had before. The next week, we quantified the DNA by using the PCR amplification. We used the 16S rRNA ribosomal RNA gene, and once we figured out the concentration of the DNA, we used the absorption spectrophotometer to do this. Your DNA has a 260-280 ratio, so this is what we were looking for when we were trying to see with the absorption spectrophotometer. And we used the PCR to find the V3 and V4 regions of the DNA, and we did this at 460 BP after amplification, and we were expected to have 550 BP. So we used the nano drop spectrophotometer to also test the concentration again. Then we used the thermocycler for 25 cycles. Then we completed for the PCR amplification. And then the next week we completed our gel electrophoresis, which you can see is up here. And we used this to separate the DNA fragments from different sizes. We set the power supply to 250 volts, and after this was complete, we turned on the light to see the bands in the picture, as you can see here. After that, the next week, we used the Engine Quart Ampure XP kit to clean up the amplification reaction, and this removed all components of the amplification buffer, like Mg2+, enzymes, proteins, and the unused old primer that we didn't need anymore. The reason we removed these impurities was because we didn't want any cross-contamination with the DNA indexing, because that could affect the success of the process. And then we did DNA indexing, and we did this to amplify the regions, and we found the nucleotide sequence of the DNA, and We did 8 cycles instead of the 25 cycles we had done before, and we did them at 95 degrees for 30 seconds in Celsius, 55 degrees for 30 seconds, and 72 degrees for 30 seconds. After we were completed with the DNA indexing and we found the nucleotide sequences that we needed, we then did the PCR cleanup again, just as the same process as was done before. In the 5th week, we used a fluorometric assay to determine the concentration of the amplify sample after the PCR cleanup, and we needed a more precise measurement, so that's why we used the fluorometric assay instead of the spectrophotometer, because it just has a more accurate and precise reading that we needed, and that's why we used it. So after we prepared the dilutions, we used the Illumina MISSEC to next generation DNA analyzer, which is where we got all of our graphs, determine the nucleotide sequence of the indexing PCR fragment, and to be able to find the specific bacterias and how many reads we had in each bacteria. So after we were completed with the DNA indexing and we found the nucleotide sequences that we needed, we then did the PCR cleanup again, just as the same process as was done before. So for the results, the purity of both of the samples was 1.59 for sample 793 and 1.8 for sample 1048. We ended up having to get samples 365 and 406 for the data for the graphs. So the fluorometric readings and the amplification PCR readings were good. We got good results with those. We had good volumes. We ended up having to get samples 365 and 406 for the data for the graphs. You need 1,000 reads for it to be viable, usable. The male dog had 14,000 reads and the female dog had 10,000 reads. So for the gel electrophoresis, we got good reads. We were able to separate the DNA fragments from each other. And when you look at our graphs, you can see that in the dog, both dogs, it was almost 100% bacteria. I think it was like 99.7% bacteria. And for the positive control, it was 100% bacteria. And the unclassified reads are reads that are not found in the database. So it doesn't mean that they're the same bacteria that's found or that it isn't bacteria. It's just a bacteria that's not in this specific database. As you can see with the orders, basically some of them may have different amounts of different orders in bacteria, but it's all the same bacteria that we found. There wasn't much of a difference in order. So the bacteroids, the male dog had more, but it's not really a significant difference. And the clostridiums, the female dog had more, but that's just not a big enough difference for it to be significant to say that they have a difference in their gut microbiome. And the fusobacterioids, they are almost the same as the Burkholderbacteroids. It's almost exactly the same for order. For genus, there's a bit of a difference. Male dog had some unclassified and some different bacterias than the female dog, but there is some similarity as well. And we believe that in some of the bacterias that they had, the male dog may have just had in such a small amount that it wasn't significant, but it was present in the gut microbiome. So the Megamonas female dog only had that, and it was about 20% of the genus, and Fasciobacterium was only in the male dog, but it was also a small amount. So it's hard to say that just this small amount of difference in the genus would cause an effect for there to be a reason to state that the female and male dogs have different gut microbiomes. One is healthier than the other. This doesn't really give us reason to say that, which is what we talked about in our discussion. We used absorption spectrophotometry to determine the concentration of the DNA first, and we would use that to determine whether or not the samples were good and viable to use to carry out the experiment. and then that meant that we could use them if they were the correct absorption. So ratios below 1.5 indicate that it was not concentrated enough and there was not enough DNA available to use the samples. Fortunately, both of our samples fell in the correct category, and like I said before, our first sample had 1.59 and the other one 1.8. Those are both greater, so we were able to use them. And then we, in order to measure the DNA, the amplicon was present in our sample, applied gel electrophoresis to see if we could separate the DNA fragments from the sample, and we were successful with that. As you can see that in well 2, which our first sample was located, DNA is present as well as in 2, but in this well it was the negative control actually, so no DNA should have popped up. We believe that we could have accidentally loaded the well with the wrong sample, or there could have been error with cross-contamination. This part of the experiment that I will talk about next is much different than our previous plans because our data was lost after setting up our index PCR. After the index PCR, we were instructed to test and randomly choose data in order to practice fluorometric assay quantification, and the techniques of the testing concentration through UV absorption was not selective enough because it wouldn't distinguish the RNA from the DNA, which is what we needed to happen. And we measured the fluorines using a qubit fluorometer, and we had to determine whether we had a high enough concentration to be able to dilute it to 100 microliters. Both of these samples were not high enough concentration, therefore if we had to dilute it to 100 microliters, we wouldn't have enough DNA in the solution. The results used from this point forward are also from different animals than we originally started now, because the presence of unclassified organisms at the kingdom level, because this is only 99.7%, that 0.03% is unclassified, shows cross-contamination because it should be 100% bacteria. The kingdoms and orders found in both specimens were matched up very similar. As I said before, they were all almost 100% bacteria, but the 0.03% was unclassified, so that was just a bit of cross-contamination. The genus bacteria such as Bacterium, Magnomoras, Boratilla, and bacteroids were all bacteria that were common with each other. Vascobacterioids, this one right here that's only in the male, are very prominent to the male species, so that could be why it was different. And bacteria such as Megamonas, Blatulia, Fusobacteria are very prominent to females, so that is why there's a small difference there. But again, we weren't able to conclude that there was a significant difference between the gut microbiome of the male and female dogs. So when starting this experiment, we faced ourselves with a hypothesis if the sex of a canine will have a direct effect on the animal's gut microbiome. Although there are some small differences, we don't feel that it was enough to create a strong enough case to support our hypothesis in this experiment. So therefore, we must reject our hypothesis stating that there is no direct correlation between male and female gut microbiomes, and if there is a difference at all. Thank you.